So welcome everyone. For anyone that is attending this call for the first time, we host this industry-wide call every month for really any company that wants to participate in the cultivated meat value chain. And the purpose of these calls is really to introduce and welcome new solutions providers into the industry with the end goal of establishing new collaborations that are going to help accelerate R&D and commercialization in the cultivated meat field. So if your group is interested in presenting on a future call, or maybe you're working with a company that could benefit from being on these calls, do feel free to put them in touch with me directly. I'd be happy to have a chat with them. So I have a couple of housekeeping items today, but I'll first hand it over to Maddie from our policy team for a brief announcement.
I just wanted to let everybody know that GFI recently published our annual State of Global Policy report, which is a deep dive into a bunch of data and information on both public funding and regulation of alternative proteins, including cultivated meat, across the globe. So I would encourage everybody to check it out and to especially share with government relations or regulatory colleagues. And I will drop a link to that in the chat. And that's all from me.
Great, thanks, Maddie. Yeah, definitely encourage people to check out that report. And on my end, so I wanted to make a few announcements. The first is that the Good Food Conference, which is a conference that we host, is taking place September 18th through 20th in San Francisco this year. We recently released the program and the first batch of speakers. And so if you're interested in attending, now would definitely be a good time to register. I'll drop a link to that in the chat in a second. Secondly, the Tufts University Center for Cellular Agriculture's Industry Consortium is soon going to be looking for new members to join. So essentially this consortium is a public-private sort of collaboration where each member contributes a $90,000 annual fee, and that helps fund collaborative research projects that are put forth by the membership. So there's currently 9 industry members, including GFI as a member of this consortium. And so there's a lot of interesting research projects that are going on already underway. They're totally open to companies that are outside of the US as members in the consortium. So do consider this an opportunity to get involved in some interesting research collaborations. I'm going to drop a one-pager link in the chat in a second, and we'll also share additional information about this over email for you all. And then lastly, I wanted to share that applications for our 2023 research grants program are now open and are due by September 21st. And so this is for projects that are up to 2 years and $250,000 budget. And yes, we do fund proposals from companies, and we love to see companies involved in different collaborations. So we have 2 priority areas this year on cultivated meat, and I'm just going to share really briefly a quick summary of those projects on slides here. All right, so hopefully everyone's seen this. So one of our research priority areas this year is about developing tools and knowledge to promote stemness and proliferation in seafood cell cultures. So essentially, conceptually, this priority area is about improving culture conditions, cell line development, really anything about early stage R&D for cultivated seafood in particular, because as we know, a lot of the research here is sort of behind the rest of the field when it comes to knowledge for mammalian or even avian cell cultures. So this is some ideas about really what we're looking for in terms of potential applications or proposals that hit on developing serum-free culture conditions, developing cell lines that are easily accessible or can differentiate into muscle and fat efficiently, et cetera. So please take a look at our RFP, which we'll drop in the chat for potentially putting together a proposal to accelerate cultivated seafood R&D. And one of the other priority areas this year is focused on really improving metabolic efficiency of cultivated meat and also fermentation-derived processes. And so this is really about creating the data developing the data sets that are necessary to inform the creation of genome scale metabolic models, validating those models, and then iterating on those models using a variety of different techniques. So proposals here can really, depending on the organism, the cell type, or the species that you're working with, you might want to focus on upstream sort of data curation and collection. If you're working with a species that maybe already has a metabolic model, then it could be about validating that model experimentally, et cetera. But fundamentally, this is sort of the basis of helping us to improve feed conversion ratio for cultivated meat in the future, which is going to be really critical for lowering costs, as well as lowering the environmental footprint of production. So again, please encourage you to take a look at our RFP, which I'll drop in the chat, and to submit a proposal. We'd be happy to take those. And without any further ado, I'll introduce today's speakers. First, I'd like to welcome Robert Brownlee from 3D Biotissues, who will be talking about their tissue templating technology. And then at 1:30, we'll turn it over to Sasan and Gina from InSight Labs, who will be talking about their analysis platform for monitoring and optimizing cell behavior. Feel free to drop questions in the chat throughout, and we'll be happy to get to them at the end of each talk. So Robert, I will turn it over to you.
Thank you, Elliot. Can I just start by saying what a pleasure it is to be invited to speak, and hopefully you guys get as much out of this presentation, our presentation, as we have from the presentations that have gone on before. Let me just share screen. So I wanted to talk to you a little bit about 3D biotissues and what we're doing with regards to providing structure in cultured meat. What we're going to talk about today is we'll tell you a little bit about 3D biotissues, who we are, what we stand for, and then we talk about some of the challenges that the industry is facing at the moment in terms of providing structure. So ultimately, a lot of industry has solutions now for serum-free media formulations, but the big challenge is the cost, really. But also, the next stage is to look at providing structure. There's multiple different solutions towards that, scaffolds, 3D printing, etc. But we've got a slightly different way of overcoming that challenge, and that's what we're going to talk a little bit about. As mentioned, providing structure for cultured meat. We'll talk a bit about our acidity mix supplement as well, which is a big part of our solution. And not only does it drive down costs, but also it facilitates ECM deposition in cultured meat, which is critical when you're looking at providing structure and generating tissues as opposed to just proliferating and differentiating cells. Talk a little bit about collaboration partners, and then we can go through any questions that you may have in the last section. So who are 3D biotissues? Well, we're a spin out of Newcastle University, and Che Connon is our CEO, and he's also professor of tissue engineering at Newcastle University. So the company is founded and based on over 20 years of research into the space. Our vision is to bioengineer a safe and healthy future for all. And our mission is to grow truly functional lab-grown tissues by following natural cues. Now, not only are we involved in cultured meat, but we're also involved in cultured leather, and we can also culture corneas for transplantation using the techniques that we've developed. So we've successfully developed and used a range of IP-protected technologies in the production of extracellular material in cell culture, all of which produce bioequivalent constructs, the same composition and function as naturally occurring tissues such as skin, cornea, and muscle. So we don't use scaffolds, we don't use 3D printing. We're able to program the cells effectively or instigate the cells to develop the tissues themselves. Our goal is to be animal-free, ethical, and eco-friendly. So hopefully that synergizes a lot with other cultured meat companies in the industry. And this is the big challenge, really. And the next challenge, should I say, for cultured meat is providing structure. And without structure, there really is no meat. And this is what we were promised: is beautiful steaks with the bite, mouth feel, and texture that we're looking for. Whereas the reality is, providing structure is a huge challenge, and we're not there yet. There's multiple solutions, as mentioned, out there. But that alongside cost reduction is the big challenge that we're facing. So the problem, as mentioned, and stage 2.0 and 3.0 for cultured meat is creating structure from our cells. And ultimately that is tissue engineering. So it's not just about proliferating and differentiating the cells, it's about providing the tissue organization. And tissue structure is incredibly complex. So from nano through to macro scales, and hierarchical organization is essential to all tissue structures. Interestingly, appropriate structure provides proper function, and with regards to cultured meat, that provides texture, bite, and mouthfeel. And the feedback from the market currently is that the current products have a very soft texture and structure and mouthfeel, similar to tofu, which is different to what we're trying to achieve. And accurately replicating nanoscale organization of tissue ultrastructure is a considerable engineering challenge. And this is our solution. So what 3D biotissues do is we use the inherent power of the cells to engineer the tissue. We provide nanoscale resolution and hierarchical structure and physiological function. Now that's applies to our other products, but in terms of cultured meat, that provides a bite and mouthfeel that is desirable, but also nutritional value. So our tissue engineering platform that we've developed, we apply physical and biochemical cues to direct and organize the cell growth. It uses the inherent power of the cells. And self-assembling cells form highly structured natural tissues. A big part of this is the ECM deposition. Now, ECM deposition is a considerable challenge for cultured meat. Serum-free culture, it is difficult. And a big part of our solution involves the increased ECM deposition that we get from using our CityMix product. So our techniques, we use active and passive cues for controlled tissue organization. Methods to increase tissue volume and function, and as mentioned, CityMix is a big part of this. And a modular approach to complex tissue. So we can create these meat fascicles in these modular formats, but we can also layer our cells and tissues if that is the desired outcome. So it's engineered by biology. And with our product, you see fine-tuned nanostructure. And interestingly, with the increased structure, the knock-on effect is we see increased biomass, as evidenced here in the structured biomass and the random. And ultimately, this is what you get. So our cultured meat is formed by fibrillar tissue, which is exactly what we're looking for from a texture, mouthfeel, and bite perspective. And that is comparable with that of natural meat. And you can see the fascicles and the muscle fibers here in natural meat. So now moving on to CityMix. So as mentioned, CityMix is a product that we're supplying to the cultured meat market currently. And it's essential for providing structure without a scaffold via increased ECM establishment in the presence of ascorbic acid. Now, we know ascorbic acid is very, very important in serum-free media in order to provide the raw materials for the cells to deposit ECM. And not only does it facilitate greater ECM deposition, but it also has a remarkable effect on lowering growth factors in your media. It's a patented non-toxic formulation, so there's zero toxicity. And this is probably the most important line in the presentation. It's inert. So it doesn't react chemically. It doesn't act as a growth factor. It doesn't act as a nutrient. It doesn't act as a food. But it's inert, food-safe, high molecular weight molecules that facilitate macromolecular crowding. Now, there's quite a bit on macromolecular crowding in the literature. And what macromolecular crowders do is they have an effect on increasing biomass production, and they significantly reduce your recombinant cytokine requirement. Now, recombinant cytokines represent a huge cost factor for serum-free media in the cultured meat market, and we see in the presence of city mix, an ability to reduce the recombinant cytokines by 50 to 75% without any effect on yield. It significantly reduces recombinant albumin requirement, and according to the Good Food Institute, recombinant albumin is going to be one of the #1 cost concerns in terms of serum-free media for cultured meat. So what is it? Well, we provide it as a supplement. It's a 25 times supplement that you add directly to reduce or replace serum proteins and our synthetic growth factors within established media. And the benefits, as mentioned, include 50 to 75% reduction in recombinant cytokines, a 75% reduction in recombinant albumin. We see increases in tissue volume. It's totally animal-free. We have not seen any bioaccumulation in biomass, and it's chemically defined and food safe. It's currently used in the food and cosmetic industry. So there's E-numbers associated with it. We eat it all the time. So from a regulatory perspective, we don't envision any issues there. And because it's chemically defined, we can make a lot of it. So we've actually done modeling to see if we can provide city mix for the entirety of the current meat, current natural meat industry, and we can. So there's the continuity of supply is not going to be an issue. So now let's move on to some of the data on reducing media components. So this is some data that we generated in Cultivated Media, which is a well-established media in the literature. And this is on the proliferation of immortalized and primary porcine cells in reduced supplemented serum-free media. Now, we did this in myoblasts, adipocytes, and fibroblasts. And what we did was we reduced the growth factors by 50% in Cultivated Media. So we reduced human albumin, IGF-1, FGF2, and HGF by 50%. And you can see the cell number associated with the reduced serum-free media. Now, by adding city mix alone to that media, we saw 1.4 times increase in cell number. In terms of pre-adipocytes, fat cells, obviously notoriously difficult to grow in serum-free media, and it's a big challenge for the industry currently. By adding city mix alone to that reduced media, we saw a 1.6 times increase in cell number. And with regards to fibroblasts, we saw a 1.3 times increase. Now, these were cultured for 7 days, and that's with a 50% reduction in growth factors. So when you think about the cost of your serum-free media currently, a 50% reduction in growth factors represents a significant cost saving. And that ultimately just reduces the cost and brings us closer to meat price parity, which is ultimately the goal here. Now, this is some data on the BF9 media, again, a very well-established media in the literature. And this is in porcine myoblasts grown for a 7-day period. Now, what we did here was we reduced the growth factors by 90%, just to get a baseline. And you can see we don't quite have equivalency here to the control, but you can see there's a 1.8 times increase in terms of cell number in that reduced media and adding city mix. So now, if anything, 90% reduction in growth factors was too much because we haven't got equivalency here. So we generated additional data on this on BF9, and instead of reducing by 90% the growth factors, we reduced everything by 75%. And you can see the levels here. And again, this is porcine satellite cells. This is grown for 3 days. So we reduced insulin, transferrin, selenium, FGF, NRG1, TGF-beta, and recombinant albumin by 75%. So you can imagine the cost reduction that we're seeing here. And you can see there's no significant difference between the reduced media, including city mix, and the full media. We also carried out a similar experiment in the Essential 8 media, again, a very well-established media for growing cells. Slightly different formulation. It has FGF2 and TGF-beta1, but we reduced them by 75%. And you can see this was after 7 days. Again, porcine satellite cells, and you can see the equivalency here in terms of cell number, percentage cell number. So we also wanted to get some suspension culture data as well, and this was actually done by an external lab. external cultured meat company. And what they did was they reduced their EGF and IGF-1 by 75% in suspension culture. And you can see there's quite a bit of variance in terms of the 100% growth factors, but you can see excellent results with a 75% reduction in those cytokines in the presence of CityMix. And the knock-on effect of this is just a massive reduction in your media costs. And we know that media is a huge concern in terms of getting price parity. So now moving on to ECM production, and ultimately when we're trying to make stable tissues, ECM production is of fundamental importance. And this is data in human dermal fibroblasts. These were cultured for 14 days in DMF12 glutamax supplemented media with ITSX, ascorbic acid, crucially, because we know ascorbic acid is very important, and 5% FBS. And you can see in terms of collagen 1 deposition, the presence of CityMix increased the collagen 1 deposition by 5 times. From a collagen 3 perspective, we saw a 24 times increase in collagen 3 deposition. A decarin, that's a 14 times increase in decarin, and also a doubling of elastin. So incredibly powerful results in terms of the cells depositing their extracellular matrix in order to improve the tissue formation. So there's a real boosting effect that we see CityMix adding to these media. This is some data in murine myoblasts, and this is so C2, C12 cells cultured for 5 days. And you can see here that MyHC is a late stage differentiation marker. And the problem with serum-free media currently is that it rapidly increases differentiation, creating unstable tissues. And you can see here that serum-free media plus CityMix stabilizes the differentiation to that of your standard differentiation protocol, which is 5% FBS. And alongside that, you can see the fibrillar collagen deposition. And the imagery here is a good example of the density and stability of the cells and tissue there, and also the unstable tissue in the serum-free media. So incredibly powerful results in terms of tissue formation and stabilizing differentiation. So what does that mean? Well, so in terms of working together, 3 biotissues are not manufacturing meat currently. So there's no conflict of interest with other cultured meat providers. What we're doing is we're investing in solutions to the industry's problems and not focusing on meat production. We know there are a lot of other companies in the space that are much farther down the bioprocessing and manufacturing route. And what we're looking to do is work with these industrial leaders on their structured product offering. We can license our technology, both CityMix and the tissue templating technology. And CityMix is available now. So if you're advanced in your manufacturing bioprocessing, if you've got your serum-free media developed and you're looking to take your cultured meat to the next stage, CityMix can reduce your cost of manufacture. The beauty about it is you don't need to go back into your R&D to reformulate. You can just take your current formulation and lower the cytokines and the albumin by 50 to 75% and use the same thing. You can also combine your cells and manufacturing with the 3D biotissue structure technology. So we're very open to discuss collaborations for mutual benefit on a combined structured product with all cultured meat companies out there. So, and this is my Mary Antoinette reference. So let them eat steak. Ultimately, that's what we want them to do. We want everybody to be eating cultured steak at some stage. And this is an example. So I'll finish off here and take any questions. But this is an example of our pork fillet that we made and developed. And you can see the structure there. Yeah, it has the fascicles, and that is without a scaffold and due to the templating process. And so that's everything. So I'd like to open the floor to any questions.
Great. Well, I'm going to cut off the conversation just in the interest of time for our next speaker. Rania, if you have any questions, you can drop them in the chat, and maybe Robert or Ricardo can answer those in the chat while we let pass it on to the next speaker. Thanks. Should probably be relevant for this cell monitoring conversation. So Seshan, over to you.
Thank you very much, Elliot. Hello, everyone. It's a pleasure being here, and I'm very excited to give this presentation today because I'm a huge fan of a lot of what you guys present here are working on, and I'm very much looking forward to this interaction. All right, hello again, everyone. My name is Shoshana Gina. I am the co-founder and CEO of InSight Labs. And the topic of my presentation today is how single-cell biology can be used for biomanufacturing. And the reason why this is an important topic is because as a methodology, it gives you incredible insights into your experiment or biomanufacturing process. And it also significantly improves your productivity overall as a team. So before I get started, I did like to mention two things real quick. The first is, yes, this is a presentation, but I hope to make this as conversational as possible. And I guess what I'm trying to say is I'm looking forward to your feedback and questions at the end of the slide. And the second thing is just to do like a quick overview about the structure of the presentation. The first part, I'm going to be talking a little bit about who we are as a company and some of the challenges within the cultivated meat industry as it relates to cell monitoring and assessment. And on the second part, I'm going to be talking a little bit about our journey of discovery as a team or as a company, why we are all in this together. And by this, I mean everyone in here in this room and in the community at large, and also say a little bit about some of the new stuff that we're working on. So we are InSight Labs, and we like to think of ourselves as citizen scientists. As the description, we are citizen scientists. And fun fact, if you type in citizen scientist into image generator, like DALL-E, you get interesting prompts. That's what this is about. So according to the internet, this is what a citizen scientist looks like. But essentially, we are a tight-knit group of biologists and engineers. And our goal as a group is to use our knowledge and expertise collectively to solve important problems in biomanufacturing or bioengineering in general. In the spirit of helping each other and solving each other's problems, we've spent a lot of time with our biologist colleagues and as engineers, right? And we observe how they struggle with some challenges when they're involved with lab work. And the most self-evident of all of this problem is the fact that there's a lot of drudgery involved in running biological experiments. I mean, if you look at the image here on the left of your screen, it's kind of like the classic NTT assay, right? I'm sure a lot of you guys are familiar with that. And this is, it takes a lot of steps, it takes a lot of time, and most of the time and energy of all of this really smart scientists is spent on tedious and repetitive tasks. The second problem we see is population averaging assays just aren't that helpful. They give you like an overview of what's going on, but they tell you nothing about the intricate details, which is exactly what you need if you are in an accelerated development environment. And It's also not a high throughput or high resolution system. And this means you're going to be wasting a lot of time and you'll miss a lot of windows where you have to make an intervention for your process or experiment. The third thing we noticed is that it's just about the nature of biodata. Biodata is unstructured by nature because you're collecting information from different sources. And what this means, if you don't have structured data, is you can't learn, right? You can't identify patterns, and you can't apply any kind of time series of methodology or historical methodology to see how you can improve on your process. So as a team, again, we all came together and came up with a bunch of ideas. And the best solution we could come up with is using a combination of microscopy and AI to build a high throughput system for quantitative cell analysis. If you look at this image on the right, it's a well plate, a 96 well plate that's loaded in a chamber of a fluorescence microscope. And in that well plate, you have 6 experiments that's duplicated in multiple wells. And by the way, that's all the setup you have to do. And by applying time-lapse microscopy, you can run multiple assays on multiple experiments and get your results in very, very detailed manner. So what are the obvious benefits of applying this approach? The first obvious one is you're automating all of your cell line processing and cell analysis activities, and you can free up all of this really smart people you have so they work on high value tasks like optimizing your experimental protocols as opposed to getting bogged down with assay implementation protocols. On an average, what we've seen so far is teams that adopt this, you save over 50 hours a week in your lab. The second benefit is you get a single cell report on every cellular activity. You don't miss any important event that happened in your process or experiment, differentiation, cytotoxicity, lineage, morphology. You know exactly how it happened and when it happened. And that's a very, very important detail. And this way you understand how individual cells move, activate, interact with each other, or just simply die. The third benefit is you have ability to make trained models. Because your data is now structured and it's time series, you can actually build training models that you can apply to optimize your process going forward. So I will spend a little bit of time here to say more about single cell biology. As a methodology, it's kind of been around for about 7, 8 years. And it's simply the application of technology that enables multi-omics analysis of single cells. And what that simply means is instead of just getting population averaging results, you're looking at each cell and observing a variety of property of each cell over time. And if you look at this image on the right, you would see like cells in a culture, which is this on the left, and you see a table here on the right. Every cell in this image is a row on this table, right? So you have the frame and you have a large data set. And every row here has about 27 columns. Each of those columns is a cellular property. And right here, you could see the area and the B box area. They're about 25 more of those properties. That includes the mean intensity, elasticity, the speed, the volume, the thickness, and these are all very important properties that you can use to build your own training model, like I talked about before. And single cell biology as a new methodology has provided groundbreaking insight into metabolic pathways, helping identify new cell types and molecular pathways as well as various physiological processes and pathologies. Now I would like for us to run a little bit of a thought experiment, right? Let's imagine that the image I showed you in the previous slide, right? It's a frame from an experiment, that's a single frame. Now imagine if you capture that single frame every 5 minutes for 24 hours, you would have 288 frames, right? And if you stitch all of those frames together, right, you have a time-lapse movie of your experiment or process, right? And if you also remember, each of those frames has a table, like a large data set that accompanies it. You can extract valuable information from those data sets that it's very, very useful for your process. I'm gonna play like this really short video real quick. It's of a cell, 2 different experiment that's been run in different media formulation. And I'm sorry. And if you just by a visual observation, you could see how the cells are moving on the screen, and you could see the colors. They have different colors. Each color represents a morphological status. So just by watching a simple video, you can get a very good feel of what's happening morphologically. So what does this mean? We can use all of those data that I just talked about to build cell kinetic models, right? So, and what that means is, how is your cell behaving over time? And we want to quantify all of the information of how the cell is behaving over time. And why is this important? It helps you to get comprehensive and dynamic analysis of your cell as a function of time, which is kind of just a different way of saying that you know what happens when it happens, right? And secondly, you can use all of this immense amount of data to build training models, like specific training models for your assay, like a customized training models for your assay that you could use to run multiple assays on the same experiment, or compare historical series of experiments to see how you are doing over time. Now, I'm gonna spend a little bit of time again to just say, talk about some of the new things that we're working on, because we're a startup and it's inherent in our nature to kind of push the boundaries of what's possible. We've been interacting with a lot of users, and thank you to some of the users in the community that have given us feedback. And some of the feedback we've gotten is what's served as a direction on what we should be working on. Spatial distribution of cells has been identified as a very important detail. And the way the cells align, right, is a unique function. And if you can identify those patterns and add it to some of your data analysis, you can use it to actually predict tissue function. We thought that would be a very, very important feature for the community. So that's one of the new things we're working on now. The second is transcriptomics. Lots of research papers have come out recently about cataloging and sequencing RNA molecules in a cell. And we are working on a new idea of building a 4-dimensional AI image analysis pipeline that we could use to analyze gene expression on a cellular level. So to my favorite part of this slide, please let's work together. Thanks again to some members of the community who've been really helpful in giving us feedback, but we want to work with a lot more folks in the community. We're currently in beta, and we're looking for feedback to improve our interface. I'm gonna, in the next slide, I'm gonna share a link where you could actually register for our beta and just start using a platform, and we hope to get a lot of valuable feedback. The second is we build cell kinetic models, right? That's at the core, that's what we do. And the features I showed you before, those are features that we offer today. So if you have a cell line that you're working on, and it's what you use for your biomanufacturing process, we would love to build a kinetic model for you that you could use in your bioprocess. And we hope that it's something that's useful and valuable for you to help you accelerate to that desirable outcome of an awesome steak or cultivated seafood products. So finally, thank you very much to everyone. Thank you, Renee and Elliot, for putting this together, and GFI for creating a space, because I think a space where we can collaborate openly is very important. And here is my email. Please reach out to me anytime. And I'm going to drop the beta sign up link and my email in the chat. And yeah, please, looking forward to all of your questions and interacting with you guys. Thank you.
Awesome. Thank you, Sishan. Well, if not, we will we'll share these slides with the rest of the community, and hopefully you can get some folks to sign up and get some more data inflowing into your platform. So I want to take the time to thank both of the speakers today, and for everyone that showed up. Really appreciate it. And we will see you here next month. Thanks so much.
Thank you.
Thanks, everyone. Bye-bye.
Take care.
Thank you. Bye.